All right, so welcome to Powerhouse MD. In this video, we are going to be covering mitosis. So, like usual, we're going to start off with a case presentation. A 15-year-old boy is undergoing genetic testing through cell division analysis. The pathologist placed the sample under the microscope to determine how the division occurs. Upon first glance, the pathologist can visualize by karyotyping and observes the chromosomes are condensed and maximally visible. The chromosome also seemed to be aligned at the equator. Which stage of mitosis is the cell in? So a couple of things to know before we get into mitosis is the cell cycle. Following M phase, cells can continue to G1 if cells do not have any mutations or abnormalities. However, if there is a mutation or abnormalities, they are pulled off into G0, which is their So before we get into mitosis, the first thing we need to learn is the cell cycle. Following M phase, cells can continue to G1 if cells do not have any mutations or abnormalities. However, if there are mutations or abnormalities, they are pulled off into the G0 phase, which is where cells are essentially frozen and surveyed. At this point, the cells are going to be 2N, 2D. Once in G1, if there are no abnormalities, the stage varies in length due to different rates of division. These cells are going to be diploid, and they have one chromatid per chromosome. So diploid, again, still being 2N. So diploid still being 2N, 2D. DNA damage can result in increased levels of P53, which causes cell cycles to arrest or lead to the activation of a G1S checkpoint. This activates the DNA repair system or even apoptosis if the damage is severe. If there's no mutation or there's no damage, the cell may also go. If there's no damage or mutation, the cell then goes into the S phase. This is where DNA synthesis occurs. Chromatids are copied and joined at the centromere. There are 2 sister chromatids per chromosome. Next, they enter G2, which is one large cell with 2 sister chromatids per chromosome, and this cell is ready for division. Lastly, we have the M phase. In mitosis, the segregation and separation of sister chromatids occur, and the genetic composition of the daughter cells identical to the page. In mitosis, segregation and separation occur. In mitosis, the segregation and separation of sister chromatids occur, and the genetic composition of the daughter cell is identical to the parent. This is seen in somatic cells. So the first phase of mitosis is prophase. In prophase, the nuclear envelope dissolves. So you don't really see the nuclear envelope around this. Well, what should be the nuclear. So the first phase of mitosis is prophase. But in prophase, the nuclear envelope dissolves. So you don't have the nuclear envelope around what's supposed to be the nucleus. Mitotic spindles start to form. Chromosomes are condensed and are visible. These are what you see chromosomes. And chromosomes start to spindle. So you see some sort of pattern beginning to form. Now back to our case. Some key points to notice about the case is cells are visualized by karyotyping. So back to our case. There's some key components in here that will lead us to our answer. That being visualized by karyotyping, chromosomes are condensed and maximally visible, and they're aligned at the equator. So what stage is the cell in? Well, this stage is metaphase. In metaphase, chromosomes are condensed and maximally visible. They're aligned at the equator and can be visualized by karyotyping. So here they are aligned at the equator, and the microtubules are attached to centromere's kinetochores. The next phase is anaphase. So you see in anaphase that the sister chromatids start to move towards the centrioles. This is also when you have the separation of the sister chromatids. In this phase, when non-disjunction occurs, this is called mitotic non-disjunction, and we'll get into that at the end. Lastly, you have telophase. Telophase is combined with cytokinesis typically, but cytokinesis is the very last step of cytophase. In some books, you may find that cytokinesis is its own phase, but in this case, I'm putting it with telophase. So telophase, the sister chromatids move to the opposite poles. The nuclear envelope starts to form and the chromatids are less condensed. So you see the chromosomes. So in this one, the sister chromatids move to the opposite poles. The nuclear envelope also starts to form. As you can see right around here, there is starting to form the nuclear envelope, and then chromosomes are less condensed, which is why they're not as prominent, or in this case, colorful and abundant, as you see in the other histo slides. And new daughter cells are formed through cytokinesis. The cytokinesis is developing of the cells right here. So lastly, in this very short video, we're going to talk about mitotic non-disjunction. Mitotic non-disjunction leads to mosaicism. This usually occurs during embryogenesis and with somatic cells only. This can lead to two cell types spread throughout the body. As you can see here, you have one type here, one type here. So for example, in this case, some cells are normal karyotypes. That's going to be your purple. But then you also have Turner type karyotyping, so you're missing the X chromosome in the other half of the body. Normally, this patient will present as a normal, healthy female or male. Normally, because it's missing an X chromosome, this is going to be a Turner syndrome patient. Normally, this patient would appear as a healthy 46XX female. However, some parts of her body are technically Turner-based cells because they're missing that X chromosome. Usually no medical complications are noted. This just happens to be noticed on accident. In some cases, depending on where they are, however, they can affect ovary development and gonad developments like here. So that covers our topic on mitosis. I hope you enjoyed the video. Please remember to like and subscribe and look out for future videos.